2-minute anesthesia, physiology, neurophysiology, the neuromuscular junction. The neuromuscular junction is a site of communication between the motor nerve axons and the muscle fibers. The motor end plate forms the postsynaptic part of the neuromuscular junction. At the neuromuscular junction, the action potential running along the motor nerve invades the nerve terminal, i.e. the presynaptic bouton, and induces the opening of voltage-gated calcium channels. Calcium enters and binds the snare proteins around the vesicles containing acetylcholine. This results in exercitosis. Acetylcholine then diffuses across the synaptic cleft, and each nicotinic acetylcholine receptor binds 2 acetylcholine molecules. This results in a conformational change allowing sodium and calcium influx, potassium exits. This generates an action potential. This action potential is propagated down the T-tubule initially by voltage-gated sodium channel activation and then by voltage-gated calcium activation by L-type dihydropyridine calcium channels. This results in increased concentration of intracellular calcium. This triggers calcium release via the sarcoplasmic reticulum by the ryanodine receptor, allowing excitation-contraction coupling. Acetylcholine esterases hydrolyze excess acetylcholine within the neuromuscular junction. Excitation-contraction coupling refers to the sequence of events by which an action potential, i.e. an electrical event, in the sarcolemma of the muscle initiates a sliding of the myofilaments, resulting in a muscular contraction, i.e. a mechanical event. As I mentioned on the previous slide, there's an increased level of calcium. Calcium is able to bind to troponin C. This induces a conformational change, allowing tropomyosin to move out of the actin-myosin binding site. The myosin head then binds to the actin filament, forming a cross bridge. Actin and myosin move past each other. This is known as a power stroke. This results in shortening of the sarcomere and thus muscle contraction. ATP binds to the ATPase in the myosin head. The ATPase is subsequently hydrolyzed, i.e. broken down, to allow detachment of the actin and myosin filaments. The calcium then moves back into the sarcoplasmic reticulum by the circa. This is the sarcoendoplasmic reticulum calcium ATPase to await the next muscle contraction.